So in short, we are more and more commonly running into a situation that our patients run out of options and have no drugs, or combination of drugs to longer potentially be effective for them. We need new agents for these patients. Selinexor is a new class of drug, selective inhibitor of nuclear export, which is well documented to have good rationale to be used in cancer and potential for therapeutic index because of ability to retain in nucleus, for example, tumor suppressor proteins, which are critical there to allow cells to eventually undergo apoptosis. But cancer cells develop ability to exclude these proteins from the nucleus and resist apoptosis. The drug, by inhibiting the export of these proteins from the nucleus, can potentially change this survival mechanism of cancer cells, and we know it from other cancers already quite well. The drug is making headways in myeloma, and this meeting brought very important few updates in this regard. And all seem to be showing as proof of principle of role of this drug in its development. Number one, in combination with dexamethasone in quad and pentaf refractory patient, combination with dexamethasone worked in 20 to 21% of patients with reasonable extended duration of response, which I think is outstanding evidence of this particular concept of being effective and potentially helping us in the future. Two very parallel and similar reports at yesterday's session, one from MMRC, which our university led with selinexor, carfilzomib and dexamethasone, and another from Canada reported by Dr. Ballis of selinexor and bortezomib and dexamethasone, showed to some extent very similar results. And both were made based on preclinical rationale that when you combine selinexor with proteasome inhibitors, you may unravel already achieved refractory status to proteasome inhibitor and reestablish sensitivity to these agents. And indeed, especially our study showed that in patients progressing on very powerful carfilzomib combinations, 67% of them achieved partial response or better when they were treated with carfilzomib and selinexor. I would like to stress that these patients were progressing on carfilzomib at the time of study entry, including as powerful combinations carfilzomib, pomalidomide and dexamethasone. That is a very important aspect of this study. Canadian study had slightly less pretreated patients, and their response rate were in the same range, but for those who were treated with this regimen and with maybe different schedule, it appears that when selinexor is given once a week, then therapeutic index is increasing. There is preserved and maybe even improved efficacy when some of the toxicities which emerge with this drug and combinations seem to be decreasing. And the main toxicities are, for example, neutropenia and thrombocytopenia, which emerge usually from the second cycle and beyond. When we use this drug in a weekly schedule together with weekly Velcade, as it was shown, that preserves efficacy, and those toxicities appear to be much less reduced, and in fact tolerability was beyond anybody's expectations. So no question in my mind that all of these reports are bringing this drug to our attention. There is definitely plan to evaluate this drug in a randomized fashion with Velcade and dexamethasone in Boston study, and we are looking forward how this drug eventually will help us not only in relapse refractory patient, which I think is important and definitely primary goal and need, but I think it may potentially be also finding its way to enhancing activity of proteasome inhibitors and serve as this fourth partner potentially or third partner in less pretreated patients.